So we can fingerprint T cells based on the cytokines they produce, and that's the ultimate way to define their function, right? So if we're talking about the phases of the immune response to pathogen, and at all these three stages, the cytokine profiles are different, and the cytokines that the cells make are really defining what's going on in the tissue. Not even transcription factors, but the cytokines, because that's the function that's exerted, right? So if we look at the surface markers, the classic phenotyping concept of surface markers, it's only the tip of the iceberg, and the actual function can be only defined if we look at what's happening inside the cell. So the intracellular functional markers, the metabolic programs, the transcription of factors, the signal transduction and phospho cascades. For mechanistic research, this is crucial to have this defined. So we wanted to have a panel where we could define some major cytokines. So classic Th1s, classic Th2s, and cytokines that are made by multiple cells that define the pro and anti-inflammatory responses. I was like, yeah, we will make a flow cytometry panel. I'm fully confident we can do this. So we can go not really super big, maybe 30 colors, and measure the cytokines along with the surface markers. Why can be this difficult, right? So, and the result was that certain cytokines worked beautifully, but some did not. And I want you to look at this and think, when you see the paper that talks about cytokines measured from the cells, which ones do you usually see? It would be the ones in bold here, okay? It will never be IL-5 or IL-13, okay? So this forms this, let's look under the street lamp mentality where the things that are easy to measure are constantly measured again and again and again. But if we go out of that context to real life context, this becomes a real problem because we're only seeing a pro-inflammatory arm of the immune response. We're only seeing phase 1 cytokines detected, and we carry this bias into these studies where we're looking at certain aspects of disease, or we're phenotyping cell therapy products, and we still measure interferon gamma and TNF-alpha, right? So we built a panel with mass cytometry, and I mean, everything got detected beautifully. And I was shocked because, okay, these experiments were not aligned 100%, but still we used the same clones. I couldn't understand why this not works as expected. And we wanted to do a clear-cut study, so we did back-to-back analysis. We used identical antibody clones for CyTOF and fluorescence cytometry, and we had the same culture split into 2 arms. So the ones that are, expectedly working, they work both platforms had no issues with them, right? But if we look at, for example, IL-10, here we can see the pattern that is pretty typical for other things that we were troubleshooting, where the background is so strong that if you titrate the reagent down, you end up still with the background and the signal is not there. So we can get a very clean signal for CyTOF. And with the flow cytometry, we just see the background that we cannot get rid of. And with the IL-13, we see a different pattern. That was the most striking, I think, result that we ever had. So again and again, we see it. IL-13 is not detectable by flow cytometry in the hands of like 10 labs that I checked with. I mean, you can see 5 OMIPs that have IL-13, and the data look really poor. It's hard. It's hard. You cannot really see it no matter what color you use. I mean, with CyTOF, we know that there are buckets of IL-13 in the supernatant, and we can detect the cells easily, one again and again and again. We've seen it so many times now, it doesn't fail to impress me every time I see this.